Hey, I'm Sean,
And I'm Dan.
and this is our presentation on assessing the variability of motor cortex excitability using transcranial magnetic stimulation.
You may be wondering, what is TMS? Well, TMS is a non-invasive form of brain stimulation that uses a conductive coil to deliver magnetic pulses to the brain. It helps us measure cortical and spinal excitability. To break down TMS a little more, I'll talk about why it's relevant. Basically, our brain controls our body's voluntary movements by sending signals through our spinal cord to our muscles. TMS allows us to mimic these movements by directly stimulating parts of the brain that are associated with specific muscle groups. TMS can be applied to other parts of the brain, such as the cerebellum or even other parts of the body, but it's typically applied to the motor cortex to study motor function. In TMS literature, researchers often deliver a pulse and simultaneously measure a motor evoked potential with electromyography. An MEP, or motor evoked potential, is a recording of muscle activity that is elicited by stimulating the brain. An MEP measures how excitable the pathway from the cortex to the muscle is. And they're important because the excitability that they measure serves as an indicator for many different processes. For example, MEPs vary across individuals after injuries such as head trauma. There are different ways to measure MEP. You can measure the amplitude, a motor threshold, or latency.
In our studies, we looked at active motor threshold and resting motor threshold. For resting motor threshold, a signal is counted as an MEP when the EMG showed a reading exceeding 0.5 millivolts. For active motor threshold, a signal is counted as an MEP when the EMG reads over 10% of a participant's maximum voluntary contraction, otherwise known as MVC. MVC is the maximum reading the electrode will detect when a participant is pushing against an immovable object with their maximum force. Resting motor threshold measures the cortical excitability in subjects when they are simply resting their muscle. Active motor threshold measures cortical excitability in subjects when they are activating a target muscle, so their muscle is already activated and the neurons are already firing. Both measures give us cortical excitability, which can be manipulated with different variables to further probe the brain. As we increase the intensity of the stimulation, we increase the likelihood that we'll get a response, but this can vary across participants. Some people get a response with a very low stimulation intensity, and other people need a very high stimulation intensity to get the same response. We quantify this response by measuring these active or resting motor thresholds.
We had two main hypotheses in our study. First, we aimed to develop a methodology capable of gathering data that produces results consistent with intra and inter-session variability reported in TMS literature. Basically, we expected to get similar results both across sessions and within sessions when measuring the motor threshold in subjects. Secondly, we expected that the regions of interest identified using neuronavigation technology would produce more reliable results than non-neuronavigated approaches. This study will have implications for future TMS research. It will provide clear guidelines to consistently and effectively apply TMS, and hopefully we will be able to pair a task with TMS in future studies.
Now taking a look at our methodology, we use the DuoMag TMS equipment and the BrainSight neuronavigation technology to conduct these studies. First, we had to calibrate the DuoMag TMS equipment with the BrainSight neuronav so they could work together. The approximate location of the motor cortex was then located on the participant, and the approximate location of the region of interest we wanted to simulate was identified. The motor cortex is on the posterior side of the frontal lobe, which can be seen in this figure right here. Once the approximate region of interest is located and the head is properly calibrated, the coil should be positioned so that the crosshairs are over the region of interest with the coil angled at a 45-degree angle in the anterior to posterior direction. This ensures that the cortical excitability is directly stimulated by the coil. The intensity of the first TMS pulse should be low, approximately 30 millivolts to start, especially if the participant's motor threshold is unknown. If no MEP is elicited, the intensity can be gradually increased, or the coil can be repositioned slightly to try and elicit an MEP. This process is repeated again and again until an MEP is elicited. Once we have the MEP, we are able to sample the region of interest and mark it for repeated application. Once the approximate hotspot is identified, we systematically move the coil around the hotspot to see if a larger MEP can be elicited. If the region of interest we originally identified is what produces the largest MEP, we mark it as the hotspot. If a spot in the surrounding area causes a larger MEP, we sample it and then repeat the process, systematically moving around that spot to see if we can find a better hotspot. Once the best hotspot that produces the strongest MEP is identified, we are then able to use the motor threshold assessment tool, MTAT 2.0, for identifying the participant's motor threshold. For resting motor threshold, participants rest their hand on a cushion, and for active motor threshold, the participant maintains a constant force against an immovable object. The results are collected multiple times across multiple days to assess intersession validity.
In this video, I am shown on the right applying magnetic pulses of TMS to Sean's right motor cortex. I'm holding a TMS coil in my right hand and steadying it with my left. To the left of the camera, not pictured, Sean is wearing an EMG electrode on his left FDI muscle on his hand. To the right, the neuronavigation screen can be seen, which shows me where to apply the coil. To the far right is a screen that displays the motor evoked potentials that are read on the electrode located on Sean's right or left FDI muscle.
In this slide, we look at an example of a resting MEP. As you can see, at this line right here, this is where the target onset occurs at approximately 100 milliseconds. And then the MEP can be seen approximately 20 milliseconds after. For it to be quantified as an MEP, it must exceed these threshold lines, which is, again, the 0.5 millivolts.
This image portrays the EMG recording produced during the recording of a subject's active motor threshold. Again, signal onset occurs at 100 milliseconds, and the motor evoked potential can be seen after a latency of 20 milliseconds. Unlike the previous figure, however, there's a lot of electrical noise preceding and succeeding the motor evoked potential. This is because the subject is pressing against an immovable object to activate the target muscle, thereby leading to neuronal activity.
In this slide, we look at figure 1, which shows us the variation in resting motor threshold measurements for 4 different participants across 4 sessions during a 2-week time span. The interclass correlation coefficient test produces a value of z equals to 0.691. This shows that there's moderate reliability across our sessions, which supports that we have moderate intersession reliability.
Figure 2 depicts the variation in average motor threshold measurements when participants were and were not wearing a Lear swim cap to control for the confounding caused by hair. 2 sample t-tests resulted in a t-value of negative 0.18 and a p-value of 0.863. Therefore, the results were insignificant.
In this last figure, we look at the intensity needed to elicit an MEP in resting motor threshold versus active motor threshold tests. As you can see in the blue, this is the participant's resting motor threshold on average, and in the red is the participant's average active motor threshold. The intensity needed from the TMS decreased from each trial or decreased when looking at resting motor threshold to active motor threshold. This is consistent with the literature, as the neurons for the muscle are already firing during the active motor threshold condition, so it makes sense that a lesser intensity is needed to elicit the MEP.
As seen in figure 1, there's no clear difference in the motor threshold results for participants across the 4 sessions. This helps to validate our intersession reliability. As seen in figure 2, there is no clear difference between the results produced using the LyraCap versus the neuronavigation technology. This shows either the LyraCap or the neuronavigation system can be used to produce reliable results, although further testing needs to be done to assess whether using both methods together produces more accurate results. Lastly, as seen in figure 3, the active motor threshold is lower than the resting motor threshold for each participant, which is what we expected.
In conclusion, using the BrainSight neuronavigation technology, we were able to produce reliable and valid results in intersession studies that showed little difference across these sessions. Either the LyraCap or the NeuroNav was able to be used to produce similar results, which oppose our original hypothesis that the NeuroNav technology would be more reliable. Active motor threshold was also shown to be lower than resting motor threshold, which validates our hypothesis. And it was significantly lower in participant 2. We need to look more into why there's a larger decrease in TMS intensity needed to elicit a response for MEP in that participant versus other participants. For our future areas of study, we like to assess intra-session variability, which we have not been able to do up to this point. We'd like to develop a consistent TMS protocol to find sicky, licky, cortical silent period, and other TMS-related information. You can see an example of a sicky or licky intracortical inhibition right here. We'd like to develop a methodology to incorporate these tests as well, now that we have a way of reliably getting results using the TMS technology. We'd like to fine-tune our data analysis of MEPs, and we want to pair a TMS task or paired TMS to a task to further probe motor excitability. Lastly, we would like to study the motor cortex excitability of subjects with or without confounding variables such as caffeine, history of concussions, and sleep loss.